Welcome to STEM Talk.
STEM Talk.
STEM Talk.
STEM Talk.
STEM Talk. STEM Talk.
Welcome to STEM Talk, where we introduce you to fascinating people who passionately inhabit the scientific and technical frontiers of our society.
Hi, I'm your host, Dawn Carnegie, and joining me to introduce today's podcast is the man behind the curtain, Dr. Ken Ford, IHMC's founder and CEO Emeritus, who is also the chairman of the Double Secret Selection Committee that selects all the guests who appear on STEM Talk.
Hello, Don. Great to be here.
So today we have Dr. Tommy Wood, a neuroscientist and frequent STEM Talk guest, and also sometimes co-host, who joins today on the eve of the publication of his first book, The Stimulated Mind, Future-Proof Your Brain from Dementia and Stay Sharp at Any Age.
That's easy for him to say. Tommy is an associate professor of pediatrics and neuroscience at the University of Washington, where his laboratory focuses on brain health across the human lifespan. Tommy is a wonderful colleague and a good friend, and he's also a visiting research scientist here at IHMC.
So today we talked to Tommy about his mission to dispel the myth that the brain is doomed to decline with age. And his book, which is available for pre-order on Amazon, offers science-backed strategies to enhance mental sharpness and prevent cognitive decline, including Alzheimer's disease. And there's so much to unpack with Tommy about his book that we decided to split his interview into two parts. Today we talked to Tommy about the history of neuroscience and how researchers go about studying the brain. And we also have a conversation with him about what is holding us back in terms of addressing an ever-increasing burden of cognitive and mental health disorders that we're experiencing not only here in the U.S., but also around the world. So as noted, Tommy's lab at the University of Washington focuses on brain health across the lifespan, and he conducts research that focuses on ways to treat a range of brain injuries, including babies who are born preterm and adults who experience brain trauma. And he also investigates lifestyle factors that affect long-term cognitive function and increase the risk of dementia. So Tommy has a bachelor's degree in biochemistry from the University of Cambridge, a medical degree from the University of Oxford, and a PhD in physiology and neuroscience from the University of Oslo.
Before we jump into our conversation with Tommy, we have a little bit of housekeeping. First, a big thank you to everyone who has subscribed to STEM Talk, and extra thanks for all of you who have left those wonderful 5-star reviews. If you're new to the show and you like what you hear, please consider subscribing. And if you enjoyed today's episode, we'd love for you to take a moment to rate the show and maybe even leave a review. It really helps us reach more people who might enjoy STEM Talk.
As always, the Double Secret Selection Committee has been hard at work, scouring iTunes and other podcast apps for the sharpest, funniest, and most lavishly praise-filled reviews to share on STEM Talk. If yours makes the cut and you hear it on the show, send us a note at stemtalk@ihmc.us to claim your very own official STEM Talk t-shirt. Proof that the committee does, in fact, exist. So today, our winning review is posted by someone who goes by the moniker ISA5511, and it reads,
I really appreciate the interview with Michael Griffin and Lisa Porter. It helped me understand why NASA and our space program are so important in today's world. We need to stop dragging our feet and get serious about space again, like we were during Apollo.
Well, thank you so much, ISA5511, and thanks to all of our other STEM Talk listeners who've helped STEM Talk become such a great success.
Okay, and now on to part 1 of our interview with Tommy Wood.
STEM Talk.
STEM Talk.
STEM Talk.
STEM Talk. STEM Talk.
Hi, welcome to STEM Talk. I'm your host, Dawn Carnegie, and joining us today is Tommy Wood. Tommy, welcome back to the show.
Thanks so much for having me. As always, it's a huge pleasure to be here with you.
Absolutely. And also joining us is Ken Ford.
Hello, Don, and hello, Tommy.
So, Tommy, first of all, we're going to say congratulations on your book, The Stimulated Mind, which is coming out March 24th. I imagine that you're very excited about this. So what gave you the idea to write this book?
Thanks. Yeah, very excited. It's dominated my life for a little while now, as these things tend to. I think I'd always really wanted to write a book of some kind. As academics, professional writers half the time. It's a very different style of writing, but it's something that you have to practice and get good at, or at least, spend a lot of time doing. And I actually started a lot of the work that I do in, communicating science and health through writing. So I started a blog during my PhD that was sort of where I kind of got into some of these areas. And so I always thought that, maybe I would turn that into a book of some kind. But for a long time, people kind of discouraged me from doing it. One of the main reasons being that once you write a book, it's fixed and science continues to change. And people sort of warned me that you'll write something and then it's going to be out of date 6 months later. And actually 6 months later is when the book comes out because you finish writing it. And then it goes through months and months of editing and processing and getting ready for printing and all that kind of stuff. So that had kind of originally held me back. But the more time I spend working in different areas related to human health and performance. So people may, if they've listened to previous episodes of STEM Talk, they may know that my main job is running a basic neuroscience lab at the University of Washington. So we try and find ways to treat acute brain injuries in babies, in adults who experience brain trauma. And then I also work with elite athletes. I've spent a lot of time working with like general population with various sort of health concerns. And then all those ideas kind of come together, I think, or they came together and made me think more and more about long-term cognitive function and cognitive decline, and therefore dementia becomes a part of that. And across all those different areas, I kept seeing the same themes and ideas in terms of what's required to support brain health and cognitive function, regardless of the scenario. And so I got to the point where I felt confident enough that I could write something that would at least have some staying power. It was going to be relevant for a good period of time after I finished writing it. And related to that was the fact that I couldn't really think of a book that I could recommend to people when they ask me questions about, how do I, support the health and function of my brain? I'm worried about my dementia risk. Is there anything I could do about that? Because the majority of books are either very technical and not particularly practical, or they oversimplify ideas, or they just focus on one area. So I try to kind of find a balance between those things. And I can't say I've solved all those problems, but that was kind of the goal, was to have something that I could give to friends and family and people that, people who asked about some of these ideas about how we can support the health of our brains, so they could have an evidence-based, but also practical sort of handbook for the things that I think are going to matter the most.
Well, those are all great reasons to write a book. And as you noted, this is pretty time consuming. And we understand that writing and editing this book consumed your life for about 18 months. And some people we know, after writing a book, talk about how hard that was, and also that they never want to do it again. So what's your take now that you're finishing it up and getting ready to release the book?
Yeah, how I feel about it has actually changed since writing the book a little bit. When I've heard other people talk about the process of writing books, and they say things like the worst thing you can do for your health is to write a health book, because you spend all your time sat in a chair, stressed about it. You forget about all the other things that we know are important for health because you're so focused on the book. And I think most of that I managed to navigate okay. I definitely had to call in a lot of favors and use up a lot of goodwill that I'd accumulated over the years by being a responsive and collaborative colleague, and sort of disappeared into my own little world for a few months. But the process of writing the book, I really enjoyed. You just get to sit there and read interesting science and think about it, and just have sort of one main focus and goal every day. And that's kind of rare nowadays with sort of the ways that we have to do work and we're pulled in multiple directions. So that whole process I actually really enjoyed. What I've enjoyed less is other than, of course, coming on STEM Talk and talking to you guys about it, what I've enjoyed less is the selling of the book, right? You write the book, but then if you want people to read it, you have to go out and tell people about it. And so that's required a lot of travel and stuff like that is takes its toll. And again, as an academic, we're not really used to going out there and selling ourselves in that way. So that's been an interesting experience. But overall, I have to say it's been a fantastic new set of experiences and skills that I've had to develop. So generally a positive experience overall.
Yeah, that's great. And so you started to allude to the theme of the book, and this is kind of, just to give our listeners a quick intro. Is it fair to say that the overarching theme of your book is that what we thought we knew about the adult brain being quote unquote fixed or static after development and then declining over time is almost certainly wrong? Is that the overarching theme of the book?
Yes. that really is kind of the core idea. And I think it's important to start with that core idea because we're at a point where we know a lot of things about how we can improve or maintain cognitive function and slow cognitive decline. But in order for us as a population, but also as individuals, to engage with that information, we have to know that it's possible for that to happen, right? If we don't think that it's possible to change cognitive trajectory, then we wouldn't bother engaging with the things that could change cognitive trajectory. So the most important idea, I think, is that just like knowing that this is possible. And these misconceptions that we have about the adult and aging brain, I think, are really kind of baked into some of the classic facts and things that we know or thought we knew about the brain. So just this idea that the brain finishes developing sometime in our mid-20s to early 30s, which is true, but we then automatically assume that because it's finished developing, that means it's no longer capable of changing its function or improving its function in certain areas. And those things are not mutually exclusive, right? So just because you've sort of like reached some point of final development doesn't mean that the brain still isn't capable of adapting or changing its function. And this then also feeds into the idea that we have about the aging brain and how the body's function changes as we age. And in the book, I lay some of the blame of this idea to somebody called Sir William Osler, who was a Canadian physician. He was one of the founding physicians of Johns Hopkins Hospital, and then a professor at Oxford until his death in the early 20th century. And Osler is credited with popularizing the idea of retirement. And the reason that he did this is because he felt that by the time humans reach the age of 60, and there's this address that he gave at Johns Hopkins, I think in 1904, where he talked about this, that once humans reach the age of 60, they're essentially useless and they need to be put out to pasture. And therefore, right, they can no longer be in the work environment because they're just like, they're no longer capable of the necessary functions. And I think we've sort of increased the retirement age since then, and some very interesting studies that showed that, right, at age 60, actually, the human brain is perfectly capable of high levels of function. But we've kind of baked these ideas into our society. We expect significant decline, at the sort of like 5th, 6th, 7th decade. And because of that changes how we approach and engage the different activities that could actually slow or prevent that process from happening. And it's also compounded by the way that we do epidemiological studies about cognitive function. So when people may have seen a standard graph of, on the y-axis, on the side, you have some cognitive function. And on the x-axis along the bottom, you have age. And you kind of see that cognitive function sort of peaks around the time that we think the brain finishes developing, sort of 20s, 30s. And then it sort of just declines from there. And this is largely the result of studies where you just take a whole big bunch of people and of a range of ages, and you just measure their cognitive function. Now, of course, on average, function is going to be lower as people get older, because there are going to be some people who lose function, and they pull the average down, right? If we're calculating a mean average, where you add everybody up and add everybody's cognitive function up and divide it by the number of people. But knowing how average function changes across a population with age is very different from thinking about how your own cognitive function will change over time. And there are plenty of studies now that show that cognitive function doesn't have to decline like that. And may even, it might be quite common that it remains stable into your sort of 70s or 80s. But we, again, need to understand that this is actually how that works. So across all these ideas, I think that we have now a lot of evidence that they're just are genuinely wrong. And one of the nicest examples, I think, that we have of this is looking at the different areas of the brain that develop the most after birth in humans, and then how that ties to how our brains age. So there have been some studies that compare the parts of the human brain that are most evolutionarily advanced, or most sort of uniquely human, so parts of the prefrontal cortex, the lateral parietal lobe, the temporal lobes, which are responsible for complex cognitive functions, decision making, language, social interaction. These areas of the brain are the areas of the brain that develop the most after birth in humans. So they're the least developed at birth. And I think this is because the brain is waiting to be shaped by the environment that it's born into. So then you spend 2 or 3 decades accumulating inputs that then shape how the brain functions. And most of that shaping happens by removing connections or neurons, right? The processes of pruning. So the brain is sort of like finished growing new cells sometime in sort of like the toddler, early childhood years. And then after that, it's refining. It's refining all the different connections based on the inputs and functions that you're sort of exposing your brain to. And this then, I think, continues essentially across our entire lifespan, because separate studies show that those same areas of the brain that are least developed at birth, that are responsible for these complex cognitive functions of humans, they are also the areas of the brain that are most susceptible to aging in humans compared to other species, like other primates. And there are some theories of the processes of aging which say that it's just a continuation of the processes of development, but there's sort of, at some point, they stop promoting growth, and that they start sort of driving some processes of aging. And I think that could make sense in that if the brain is responding to the environment and sort of pruning and shaping itself based on the inputs, if it continues to do that for our entire lives, but we stop giving it the inputs that drive development in the first place, related to how we develop complex skills, languages, social interaction, which is quite common in sort of like the normal trajectory of adult life, then as a result, the brain will start to remove functions because they're no longer being used. This is the fancy word is allostasis, where you're sort of constantly trying to match function to demand. And even if that sort of like the development theory of aging is not the true theory of aging, because there are several theories of aging, we now have lots of evidence that the adult brain, even quite late in life, is capable of developing new skills and improving function, if we expose it to the right inputs. So like the whole idea that the brain is fixed and it's only going to decline, like no matter whether you look at it from like a theoretical standpoint or a philosophical standpoint or a biochemical standpoint, everything we've learned at this point, I think, tells us that really isn't the case.
You open the book with an interesting story about barn owls. A group of Stanford neuroscientists were studying the various ways that the brain adapts to new information. This was back in the 80s and 90s, and the researchers outfitted these unfortunate barn owls with glasses that contained prisms, and these prisms redirected light so that everything around the owl appeared in a different place than it actually was. So the owl might hear something on its left, for example, but when the owl looked to the left, nothing was there. In other words, these glasses created a mismatch in the owl's brain between information from its ears and its eyes. The researchers, however, found that while young owls adapted very quickly to the glasses, adult owls did not. Even months later, the adult owls would still miss a mouse that was right under the owl's beak. This seemed to confirm that the adult brain becomes fixed at a certain point and thereby lacks the ability to adapt or change. But the researchers tweaked their study and eventually discovered something really quite extraordinary. Didn't they? Could you tell us about what happened next?
Yeah, a fascinating set of experiments that I think essentially tell you almost everything that you need to know about what is required or for the adult brain to change, or what the adult brain is capable of. And to kind of set the scene a little bit, in the original studies, you have owls, and you can find some very cute photos of these owls with they're sort of like these kind of weird rock star looking glasses with the prisms built into them. And
Elton John glasses.
Yeah, they look a lot like less colorful, but they have that sort of like they have that kind of look. In the original studies, which kind of confirmed our societal expectations of the brain, which is that the young brain can adapt and the adult brain can't. They had owls individually housed in a cage on their own, and they would just feed them. They would just provide a bowl full of food, which is essentially dead mice. And I kind of thought of this as the perfect metaphor for modern human life. You're kind of like stuck on your own in a box and like food just arrives and nothing else is required of you. And then in this scenario, the adult brain doesn't really change. It's fine. Nothing else is needed of it. No additional capacities because doesn't really need to engage with the world in any new way. But there are two different manipulations of the experimental parameters that showed that this doesn't have to be the case. So the first was when they housed the owls as a group. So they had social housing. So like all of a sudden, you're having to, well, first of all, you're interacting with your cage mates, your social group. And this requires you to know where they are in 3D space, right? So it becomes much more, you don't want to start flying into them or anything like that. So it becomes much more important to understand the world around you and then to adapt to this mismatch that's in the brain. So they found that if they housed the animals socially, they were able to adapt to the prism glasses much later in life. And another version of this is where they switched out the tasty, already prepared mouse snack dinner for live mice that ran around the cage and had to be hunted by the owls. If they wanted to eat, they had to hunt. And again, they saw that as they learned how to hunt or were forced to hunt the live mice, their brains were able to shift and adapt to the prisms. So both of these experiments suggest that the primary driver of neuroplasticity or adaptation in the adult brain is this interaction with the environment, additional stimulus, and a need for increased function, and that's what drives plasticity. If you don't have any need to drive change, the brain won't change as an adult because you're already adapted to the environment that you're existing in and you're surviving just fine. So it's this process of needing to overcome the mismatch, be it to interact with others or to hunt your dinner, that drives plasticity, but that capability is still there. And there's a nice quote in one of the papers from Michael Brainard and Eric Knudsen. And Eric Knudsen was sort of credited with sort of overseeing the majority of these studies at Stanford. And they say that a rich environment is required to reveal the full capacity for adjustment, which I think, again, essentially sort of gives us a framework to think about what the brain needs in order to still be able to adapt and improve, later in life.
This insight that an adult owl's brain is incredibly adaptable doesn't just apply to birds, but also to humans. In a study involving humans, researchers in Austria had a group of people wear goggles that flipped their vision upside down. Can you share how this study also demonstrated the adaptability of the human's adult brain?
It turns out that goggle experiments that shift how the individual perceives where they are in 3D space. These experiments have been done in humans for much longer than they've been done in owls. I think some of the first experiments were done at Berkeley at the end of the 19th century. But the ones in Austria, these were in Innsbruck, they sort of mapped how individuals adapted to a specific type of prism. So these were ones that they're sort of a complex set of mirrors that sort of flip the world upside down. And I think that the best way to kind of describe what happened is there's a quote from one of the papers that I'll read. So they say that between the first and third day, the world was upside down for the participant, as you'd expect. The participant held a cup upside down when it was about to be filled, or they tried to step over things that were on the ceiling. Swift reactions, such as parrying an attack during fencing, were in the wrong direction. But by the fifth day, the participants' clumsiness and vision started to change. Things that had been seen upside down suddenly were upright once the participant traced the shapes they saw with their hands, which required an immense effort for the brain, which they say in the quote. From the sixth day of uninterruptedly wearing reversing spectacles, permanent upright vision ensued, and behavior was perfectly correct. For example, a participant drew a picture inequality as if drawn without wearing reversing spectacles. So this is, again, in the adult brain, right, it's supposedly fixed and no longer capable of change. But within a few days, through the process of interacting with an environment or with the environment in complex ways that will sort of show the brain the mismatch between what it thinks the world looks like and what it actually looks like, that's enough to completely invert the way the world is mapped visually within the brain within a few days. But it requires, like they say, some effort, and it requires this process of doing these complex tasks. So they're talking about fencing and drawing, and they can start to figure out how this is working by sort of tracing the environment with their hands. So like starting to kind of build this new mental picture of what the world looks like. And so I think, again, this sort of shows that what we learn in owls is hopefully very relevant to humans, but it requires us to engage with the world in this variety of sort of complex and challenging ways.
These are all examples, though, in the sensory sort of domain. Have there been compelling experiments illuminating this kind of adaptation in higher cognitive functions?
That's absolutely right. When you sort of look at some of the way that neuroscience has studied develop and complex cognitive functions, vision is one of the ones that has historically been studied the most. And then, for some of these things, like for the owls, part of the adaptation also involves hearing, right, which is very important for directing the movement of the head of the owl. When you think about higher cognitive functions, it's harder to sort of completely challenge or invert the way that these are done. However, there are a number of studies with various different sort of complex skills in older adults that show or give us this idea that they are capable of change as well. So related to learning music or music theory, related to learning new languages, related to learning complex movements or physical activities like dancing, that show not only do you get improvement in either the structure or some of the sort of measurable aspects of function on imaging, if you do an MRI scan or something, that you start to see expansion or change in the related areas of the brain. But you then also see related changes in cognitive function, so particularly related to executive functions or decision-making, response inhibition. One of the sort of like intermediate studies between that, which I think, again, was one of the first studies that showed that in adults, after the brain has supposedly finished developing, you can start to see changes in the size of different areas of the brain, was in a study published in Nature about 20 years ago now, where they took individuals and taught them how to juggle. So juggling, so this was a 3-ball cascade, so like a standard juggling sequence. And they trained them for 3 months. And after 3 months, they saw expansion in the MRI in parts, mainly parts of the visual cortex. So like juggling is actually sort of a visuomotor coordination task, right? So actually a lot of it is visual still, but that you could see changes on the MRI scan related to this idea of juggling or like learning how to juggle. Then they had them stop juggling for 3 months, and then they saw that those changes on the MRI scan started to revert. So again, showing that the brain is very dynamic and it's adapting both structure and function to the skills that are being demanded of it. Then maybe a final example, which I do talk about in the book as well, because it's one of my favorite examples, is London taxi drivers, which I'm sure you've heard this story before. But there are now several studies that have looked at London black cab drivers or cabbies, and actually some parallel studies here in the US looking at taxi drivers and ambulance drivers. And the London black cab drivers are unique because in order to get a license to drive a black cab, you have to pass what they call the Knowledge of London exam, or the Knowledge for short. And the Knowledge involves memorizing 25,000 streets in a 6-mile radius around Charing Cross station, train station. And this usually takes people about 3 years to develop the knowledge. And in one of the studies of these drivers, they had 3 groups. They had 2 groups who were studying for the exam and had a control group. And these were men in their sort of 30s to 40s. And some proportion of the group passed the exam, some proportion failed, and then there was the control group. And so what they saw was that in those who failed the exam or the control group, there were no changes on a brain scan, in terms of the structure of the brain. But in those who passed, there was a structural change in the posterior hippocampus, which is important for spatial memory, which is obviously what these guys are relying on. So that kind of like showing again that if you create a new function or demand on the brain, the brain can physically adapt in response. I find it particularly interesting that while they got very good at spatial navigation, those who passed the exam actually got worse at other types of memory that didn't involve that part of the brain. And I think it's just because they diverted so much time and so many resources to that one part of the brain that then they're sort of like adapting elsewhere in response. And again, that makes perfect sense, right? The brain is trying to allocate resources to where they're most needed. But when you then look at other studies, so there was a recent study looking at different occupations in the US and risk of Alzheimer's disease or dementia. Taxi drivers and ambulance drivers had a lower risk, after adjusting for other sort of socioeconomic and other potential confounders compared to most other occupations. And similar studies in London looked at like taxi drivers versus bus drivers, where bus drivers aren't forced, right? They're on a fixed route, so they don't have to memorize and then navigate by memory, right? They just sort of follow the same route every time. And again, you see differences in cognitive function based on the complexity of the spatial navigation required of their jobs. So I think across all of that, these, as we introduce complex cognitive challenges, we see that, even after the brain is supposedly fixed, there's massive capacity for plasticity and improving function sort of directed towards those specific tasks or functions.
So, Tommy, coming back around to the overarching theme of your book, and as you've pointed out earlier, what we thought we knew about the adult brain being fixed and declining with age is almost certainly wrong. And this raises an interesting question. I'm going to say interesting, but also pretty complex. What do we really know about the brain?
Yeah, this is as much like a philosophical and existential question as it is like an actual sort of knowledge-based one. And it kind of depends on who you ask and what level of the brain you're thinking about. We certainly know some like very good high level basics. So like the brain has 100 million neurons, nearly 200 million total cells. There are nearly a quadrillion synapses that connect between those different neurons. We have a good idea about the different regions of the brain, although new regions and subregions are being found almost on a yearly basis, right? There was a recent paper that came out last year that was like, oh, hey, like these regions of the brain that we thought were sort of homogeneous actually have like a ton of different subregions within them. And we have some idea about the functions of those different regions, right? I've already mentioned some of them. But we're also increasingly appreciating that these functions aren't really located within one specific region of the brain, right? They're kind of distributed across these different networks that the brain sort of activates and deactivates based on specific functions and, rest and activity and that kind of stuff. So we have some idea of all of that, but certainly not all the details yet, right? Because we're still discovering it. But even if we think about sort of more fundamental parts, like we know how many cells are in the human brain approximately, but we don't know exactly where all these cells are or how they're connected. And we're only starting to figure that out in much lower order species. So there was a project that was run by the Microns project, run partly by the Allen Institute for Brain Sciences here in Seattle. And it took more than 100 scientists, cost over $100 million to map out all the connections in one small part of the mouse brain, in the visual cortex. And it was about 0.2% of the mouse brain. This included 200,000 cells, 500 million connections between those cells, and just storing the data just for like the structural data for this part of the brain was 2 million gigabytes of data to store it. And this is not even 1% of the mouse brain. Then if you think about scaling this up to the human brain, which is 1,000 times larger, thousands of times more complex, and then thinking about like what's the information that's being passed between these different cells, what are the connections, how are they integrating that information, which we still don't have a really good way of measuring, then you sort of quickly start to appreciate that we still don't really know a lot of what's happening inside the brain. And I think that this level of complexity that it requires to truly understand the brain, and the ways we've kind of tried to overcome that, I think have kind of held us back a little bit in terms of then practically applying this to human health and human trajectories of cognitive function, because they end up being very different things. Like one is like truly trying to understand all the different cells and genes and all those different parts of the brain versus how can I then help some individual improve their cognitive function and maintain it or prevent decline. We initially assumed that the former would lead to the latter, but at least so far there's been a bit of a disconnection there. And I think if we sort of change the way we think about that, we can maybe continue this search for knowledge about the detailed functions of and structures and how the brain kind of works, but at the same time apply some knowledge to people out in the real world that we have so far to help them. And I think we've kind of got a little stuck trying to do both at the same time. Whereas, at least for right now, they're probably sort of different pursuits.
Yeah, so you have a short section titled A Very Brief History of the Birth of Neuroscience, where you tell the story of Santiago Ramón y Cajal, who is considered the first neuroscientist. So can you, just a little bit of background here, can you tell us why he's considered the first neuroscientist?
Yeah, so Santiago Ramón y Cajal did most of his work at the end of the 19th century, beginning of the 20th century. And in 1906, he got the Nobel Prize for understanding helping to understand the structures of the nervous system. And he shared that Nobel Prize with Camillo Golgi, who was an Italian pathologist. And Golgi gave some of the first sort of structural descriptions of different parts of the brain, like the hippocampus and the cerebellum. And I think the thing that I find most interesting about this story is they were like true arch rivals. Like there was like the world's first real neuroscience feud where they refused to give their Nobel Prize acceptance speeches on the same day because they didn't want to be in the same room. But at this time, sort of this is at the end of the 19th century, the technology for microscopes had improved a lot. So you could start to magnify sections or slices of tissue like the brain and without distorting them so much that you could still see details in those structures. And the field also got better at histology techniques, so how you sort of create contrast and stain cells so you can kind of see again like the details of their structure. And so up until this point, the field generally believed in something called the reticular theory, which was that the whole nervous system, from the brain to the spinal cord, is one continuous mass and acted as such. And that latter part, I think, is actually still true. But the reticular theory was what was popularized by Golgi. It wasn't his theory, but he was the one who sort of made it more mainstream. But Cajal, sort of, looking at the structure of the brain under the microscope, showed both structurally and experimentally that the nerve cells were not continuous, but were contiguous. So there were gaps between them, right? Synapses where they communicated. And he didn't name neurons. Heinrich Waldeyer did. He sort of took Cajal's work and said, called them neurons. And this then became the neuron theory of the brain. And Cajal sort of felt that the communication, down axons and across synapses was what kind of drove the function of the brain, and then maybe explain things like learning and intelligence. And so because he was maybe the first to say that the brain was made of discrete cells that then later became called neurons, that's one of the reasons why he's considered to be the first neuroscientist.
So, Tommy, neuroscience is a relatively young and evolving field that investigates the nervous system, which includes not just the brain, but also the spinal cord and peripheral nerves. And so chapter 2 of your book is titled, How Do Neuroscientists Study the Brain? As you pointed out earlier, the human brain has something like 100 billion neurons, nearly a quadrillion synapses that control our muscles and allow us to think and make sense of the world around us. And with that many connections, it's easy to understand how complex our brains are and how incredibly difficult it is to study such an organ. And you make an interesting point, actually, we found pretty humorous, that the human brain is so complex that even such a complex organ like the human brain cannot fully understand itself. So, Tommy, can you talk a little bit about how you and your neuroscience colleagues investigate this enigmatic human brain?
Yeah. So I think the way that we study the brain, and thankfully, we're at a time when technology has really exploded, and there's so many different ways that we can study the brain at multiple different levels. And we're really scratching the surface on many of them, if not most of them. But the sort of the primary approach that's been taken to studying the brain is the one of reductionism. And this is a very common scientific technique, right? Where you have a complex system, you isolate the individual components in order to study them such that you can hopefully build a big picture and then put them back together and understand the function of the whole. And there are many ways to do this, right? A lot of them are still based on the microscope, right? That's how neuroscience began. And that's still a lot of what is done. And, this is something that we still do in my lab, right? You take some, maybe it's a piece of a brain that's, that's been taken from a human or an animal after death, and you sort of prepare it to put it under a microscope, and you can stain it with different things to kind of look at different aspects of structure. Some of this you can do in live brains or brain slices. Now you can have, like, especially in animal models, you can have like windows into the surface of the brain. You can sort of watch it doing its thing in real time, or slices of the brain that are still alive and functional, and you can kind of track those. And then there are a bunch of other techniques that have been developed, so you can measure electrical activity, and that can be at large scale or small scale. But something that's been a big part of neuroscience recently is sort of like multi-omic approaches, where you look particularly at like transcriptomics. So you look at all the different, you sort of isolate individual cells, you look at the genes that they're expressing. Maybe you dig even deeper and you look at different aspects of genetics and the structure of the different organelles and all this kind of stuff. And then, of course, you can go, to higher levels. You could do brain imaging, MRI scans. We've talked about lots of different types of scans. EEG, measure sort of networks that are active in the brain in people as they do things. But the problem that we've still had is that by digging deeper and deeper and deeper into the biology and biochemistry and structure of the brain, first of all, we haven't even like reached the bottom yet. Sort of like thinking about what we've learned about the mouse brain versus the human brain. And second of all, we haven't yet, and I would argue that on many fronts, we're quite far from being able to piece all those bits back together in order to like understand the whole. And a lot of this is probably because of this idea of emergence, which is that there are lots of complex systems where functions and capacities emerge that you can't predict from the individual components, right? You might think about different aspects of complex cognitive functions or even consciousness. And consciousness is not something I'm an expert in, but like people don't even know where it arises from or what it is. It's still argued about. But there are these things that we know about the brain that we just don't seem to be able to explain from the individual components. So I think another part of the struggle that we've had trying to turn neuroscience knowledge into practical application in terms of human health and cognitive function is that there are probably some parts of these functions that we are unable to predict from this reductionist kind of piece by piece approach. And that's where the sort of like that idea of being so complex that you can't understand yourself, even though you are that complex. I think that kind of shapes that idea that maybe we will never fully be able to do that, but we still need to make big strides in terms of human health and disease despite that.
None of what you just said is surprising from a computer scientist perspective. One can imagine somebody taking apart a laptop who didn't actually know how it works. And in the process, let's say the laptop's running some complex piece of software, even something like a spreadsheet, and you want to know how it works. And you look at all the components in your laptop. You could never find out how it works by that method. You could count every transistor, every chip, every aspect, every COM function. You would never know how it worked by looking only at the components. And so I do think that maps straight over. And your pithy observation that the human brain is so complex that even a complex organ like the human brain cannot fully understand itself really smacks of self-referential paradoxes like Gödel's incompleteness theorems or the classic "this sentence is false" liar paradox. And of course, the brain can be construed as a self-referential system par excellence. It certainly models the world, including itself to some extent. Of course, I want to point out that people apply Gödel's theorem to all kinds of things where it doesn't apply, and this is one of those things. We are not suggesting that the brain is a formal system in the sense of mathematical proofs, but It is a nicely provocative parallel, and it kind of makes a nice connection.
Yeah, I really appreciated you sort of bringing this bigger context. When I write that statement in the book, I'm actually sort of summarizing a poem written by physicist Emerson Pugh, who worked at IBM, and I think the poem wasn't published by him, but it was published by his son later on. That says, "If the human brain was so simple that we could understand it, we would be so simple that we couldn't." And this kind of, scales up and down. So you're right, these sort of biological systems, I don't think, we can't sort of directly compare over to those things like Gödel's incompleteness theorems. But I think that this idea is useful. The reason why I talk about it in the book is because I think it just allows us to maybe understand that there's so much complexity here that we can just release, maybe just start to release some of it in search of some bigger ideas that have more practical application, even if we don't really understand everything that's sort of going on underneath the hood.
Yeah, I agree. And it's a useful connection, though not actually technically parallel, in that in Gödel's theorem, the issue isn't complexity. It has to do with formality and self-reference. However, it is a nice analog in a sense. Gödel's theorem is like lots of things. It gets mostly used by people who don't understand it. So it's like quantum physics is like that. I call these people the new Mysterians. They'll evoke, it's quantum microtubules. Oh, okay. Thank you very much.
STEM Talk is an educational service of the Florida Institute for Human and Machine Cognition, a not-for-profit research organization investigating a broad range of topics aimed at understanding and extending human cognition, locomotion, health span, resilience, and performance.
Well, Tommy, to illustrate how complex this research can be, you have a humorous but insightful section in the book called "Can a Biologist Fix a Broken Radio?" So can you explain how biologists and broken radios potentially have something in common or not?
Yes, so my radio analogy is actually almost identical to Ken's laptop analogy. And so people start to have appreciated the issues that might be there. But this is actually taken from an essay written by Russian biochemist Yuri Lazebnik. He was a professor at Cold Spring Harbor, and it was published in Cancer Cell nearly 25 years ago. And it's called "Can a Biologist Fix a Radio?" or "What I Learned While Studying Apoptosis." And I read this right when I was starting as a faculty, I don't know, 7 or 8 years ago now. And I had sort of increasingly seen how my field, so this was neonatal neuroscience, had sort of moved away from the techniques and ideas that I had trained in, which is essentially you model an injury, you apply a therapy, it does or doesn't work. And right, if it does work, then you apply it in increasingly more complex models, right? So you go from rodents up to other species, sheep, piglets, and then you do a trial in humans. And like you can do all of that without really understanding sort of like the real nitty-gritty of all the complex mechanisms of what's happening. And the field was sort of moving away from that and very much going into this much more reductionist kind of approach that I mentioned earlier, right? So rather than just trying to find practical solutions to an injury, people were increasingly focused on, right, if I find this one particular mechanism in this one particular cell type, then that can become a target, right, and then sort of build from there, even though the main things that worked in this field, or the one therapy that worked in this field, which is hypothermia for what we call hypoxic ischemic encephalopathy, which is an acute brain injury that happens around birth. The babies are cooled down for 3 days. That works via a whole bunch of different mechanisms, many of which we don't understand. But that's the thing that's been most successful. But the field was still kind of thought, if we dig deeper and deeper and deeper, we can find one mechanism that we can target with one drug, and that will have an effect. And so, like, that was the context of like when I read this essay, and it had a really big impact on me. And so I'm just going to read an excerpt because I think that kind of helps. So, right, if you're imagine you're an NIH-funded biologist who needs to fix a broken radio. And so the essay goes, first, we would obtain a large supply of functioning radios to dissect. We would open the radios and find objects of various shape, color, and size. Because the objects would vary in color, we would investigate whether changing the colors affected the radio's performance. We would then remove components one at a time and catalog them. Once all the components are cataloged, the connections between them described, and the consequences of removing each component or their combinations documented, this would be the time to ask, can the information that we have accumulated help us to repair the radio? If the radio has tunable components, which is the case for all radios, and is this, especially the case for all live cells and organisms, the outcome will not be promising. Indeed, the radio may not work because several components are not tuned properly, which is not reflected in their appearance or their connections. And so when I read this essay, I chuckle because I can imagine neuroscientists like changing the colors of neurons to see if it changes their function. But in reality, this is the core problem, right? If you sort of like take something complex like a radio apart, you can't understand like how that results in its final function, just like the laptop. It's, and maybe the laptop is an even better example because you have things like complex programs and spreadsheets and things that, right, they're not, if you open it up, they're not in there, like you can't find them. And so this, I think, kind of sets the scene again for thinking about how have we historically thought about approaching complex biological systems, and why has that not translated over to a greater number of interventions or treatments for diseases or, in neurology. And so like that one that I mentioned earlier, hypothermia, I think that was the last major breakthrough in treatment for any neurological disorder in humans. Hypothermia was first described in the 1950s. It became standard therapy in 2010. And I don't think we've had anything major since. And that was probably the last major one since the 90s when they discovered triptans for migraine. Most other things done in neurology are just advancements of interventions that were developed in the like early on in the 20th century, and we haven't really, unfortunately, made too much progress since then.
Well, if you were writing the book now, as you noted, it would be titled something like "Can a Biologist Fix a Broken Computer?" Because that's much more analogous in the discontinuity between the appearance of a component and its function. And so it's a lot better connection or better analogy, I think.
So up until the 1970s, it was widely accepted that the adult brain was fixed and incapable of neurogenesis. And actually, this belief stemmed from some of the work that Cajal did back in the late 1800s. And he wrote that once the brain's development was complete, that the adult brain lost the capacity for regeneration and also plasticity. So today, we now know that isn't the case, as we've been talking about a little bit. So can you touch more on this, Tommy?
Yeah. So it's funny that Cajal, the original neuroscientist, right, came up with some of these ideas that have become fixed ever since. But he also slightly disagreed with himself because there's a well-known speech he gave at the Royal Society in London where he said the ability of neurons to grow in an adult and their power to create new connections can explain learning. So he was thinking about these ideas of plasticity even in the adult brain. And this then became the origin of the synaptic theory of memory, right, where based on experiences or knowledge, right, synapses and connections are shaped within different areas of the brain to kind of contain memories. But would also, right, if we think that's the case, then if you're still capable of making new memories, then the brain has to still be plastic. And I think that there's sort of been a fundamental misunderstanding or conflation between two different ideas that have kind of, again, held us back in terms of what we think about, the adaptability of the adult brain later in life. And so this is the difference between neuroplasticity and neurogenesis. So neurogenesis is the making of new neurons. And not much of that happens in the adult human brain. We know that. There are maybe two places where it's been described fairly consistently. So one is in the olfactory bulbs. So the sort of parts of the nose that are responsible for smell. And then in the dentate gyrus, which is a certain part of the hippocampus. The rest of the brain doesn't grow new neurons. We know that. But and so I think that because the brain doesn't develop new neurons, we assume that it's fixed, or we have assumed that it's fixed. But just because the number of cells stays the same doesn't mean that the connections between them are fixed. And so that's the idea of neuroplasticity, or synaptic plasticity, which is that the connections between cells are continuously adapting based on inputs and exposures. And that's what shapes the development of new functions, right? Even adults, all the stuff that we talked about earlier. So when you see on a brain scan that some part of the brain has increased in size or it's changed its structure, you don't need to grow new neurons for that to happen. There's a whole bunch of other things that go on. So it could be based on how those connections are arranged, but then there's a whole bunch of other cells and there's the extracellular matrix and all the things that hold the cells together. All of those things can be shaped by experience and exposures in order to support changes in function. So I think that this idea of neurogenesis ending in the adult brain has kind of held us back from appreciating how truly adaptable that the adult brain is. And it's kind of similar to muscles. So your human skeletal muscles, right, your biceps and your quads and all those, you don't grow, like when you grow the size of your muscles, you don't grow new cells. So like hyperplasia, that's called, doesn't really happen in humans. But you can make them bigger and you can make them stronger. And so, right, that's the process of hypertrophy. And you can improve the sort of the neuromuscular connection, like how efficiently they work and how they're recruited. So just because the number of cells is fixed doesn't mean that the tissue isn't adaptable. And one theory of Alzheimer's disease in particular is that it's a fundamental loss of neuroplasticity. So that would again suggest that throughout the entire lifespan, the brain remains plastic until some, number of things come together and they're lost, and then that's what drives certain sort of neurodegenerative or dementing processes. So I think just reframing that again allows us to understand that there's a huge amount of capacity for adaptation, but it doesn't mean that we're just like growing new cells to do that.
That was a nice answer, Tommy. In terms of understanding what our brains are capable of and how neurogenesis works, we need to first understand what our brains are made of. Now, I suspect that most, if not all, of our listeners are familiar with neurons, at least somewhat. These are the fundamental units of the nervous system. But I suspect that many of our listeners are much less aware of glial cells. And there are nearly as many glial cells in the human brain as there are neurons. Glial cells, which are named after the Greek word for glue, were originally thought to play a minor role in the human brain. They were simply the nervous system's glue, as it were, that allowed neurons to sort of do all the real work. But now we know the glia play a much more significant role. Actually, you argue in the book that glia should be considered at least as important as neurons. And lately I'm seeing this from various sources, a renewed interest in glia. Could you elaborate on this?
Yeah. So this increased focus on glia has become a major part of neuroscience over the past 10, 20 years. And historically, I think that people might say that neuroscience was too neuron-centric because it was sort of like the rest of the cells in the brain were generally ignored. The glia are kind of lumped together, despite the fact that the only property they share is that they are not neurons. And I think that's kind of left them behind a little bit. And this was part of the feud between Cajal and Golgi, which is Cajal was very focused on the neurons, whereas Golgi felt that the glia played an important role as well. And in reality, his idea of a reticulum where there were all these different cells acting in concert to get the final output, right? I think that is actually closer to the truth, even though Cajal was right that, yes, they are made up of individual cells that communicate with one another. So the three main types of glial cells in the brain that are very diverse, depending on which part of the brain they're in and age and species and all that kind of stuff, are astrocytes, oligodendrocytes, and microglia. So astrocytes or astroglia, oligodendrocytes are also called oligodendroglia. The astrocytes are essentially responsible for fluid flow and waste clearance in the brain, but they are also electrically active, and they connect to neurons to help regulate electrical activity involved in things like attention and memory formation. A single astrocyte, and they're called astrocytes because they look like stars, so they've got like tons of these projections that radiate out. And it's estimated that a single astrocyte in the human brain can connect to up to 2 million neuronal synapses at the same time. So, just like massive complexity in communication across 3D space and multiple cells. Then oligodendrocytes, they make up the white matter of the brain. So by sheathing the axons of neurons in a fatty sheath called myelin, you can increase the speed of conduction of those neurons. And if people think about the human brain in cross-section, on the outside, sort of like the wrinkly bits, right? That's the outer gray matter. That's your, the cortex. And there's some gray matter underneath as well. And it's called gray matter basically because it doesn't have myelin. And myelin is the bit in between. And because it has such a high fat content, it looks white. The expansion of the white matter is a critical part of human brain development relative to other species. So humans have a lot more white matter than most other species. And so like rodents have about 10%, 5 to 10% white matter. Other, sort of higher species with gyrophides, so like, those wrinkly, complex folded brains, they might have like 50% white matter, whereas humans, it can be more than 60%. And this is important for, controlling metabolism, like cell metabolism within the brain. It's important for ensuring fast connections, across networks within the brain and then from the brain to the body. I call the oligodendrocytes and the white matter the most complex junction box in existence, because that's essentially what it can look like, when you image it. And damage to the white matter is responsible for a number of neurological diseases that we only see in humans. So cerebral palsy, multiple sclerosis, and Alzheimer's disease too. And there are some data that suggests that the change in function of the white matter, or the change in structure and damage to the white matter, tracks more closely with cognitive decline and dementia risk than other things that we've tended to focus more on, like amyloid and tau. And the oligodendrocytes may be the cells in the brain that are most susceptible to damage from various places, and issues like changes to the blood supply. So they sort of like really shape part of what it means to be human, and some of the diseases we're susceptible to. And then the final group are microglia, which I think, and so mikros in Greek being small, so these are the small glia in the brain. And I think it's safe to say that the microglia are the most complex cell type in the human body, or in any body. They have, there are maybe thousands of different types depending on where they are in the brain, the functions that they're doing. And they do things like regulating the connections of neurons at synapses, regulating the formation and removal of myelin by oligodendrocytes. They regulate the production and function of blood vessels. They oversee, as sort of like the main immune cells in the brain, they oversee how the brain like regulates and responds to injury and exercise and sleep. So, there's been a huge focus as well on microglia as a potential part of all of those diseases I mentioned earlier, because they sort of seem to have this controlling process in, they have their fingers in many pies, so to speak. So like when you look across all of those different cell types and the way that they regulate neuronal function, regulate brain function, they are really having a significant impact, if not sometimes like more of an effect than the neurons are having on like how the brain is shaped and how it functions.
So that was a great overview, Tommy. And another critical component of the brain is blood supply. So the ability of our brains to continuously adapt to our environments and develop the wide range of skills that we each have is really only possible because of blood flow to our brains. And this is why cardiovascular health is so critical for our brains. Can you touch a little bit on the impact of blood flow and cardiovascular health and brain health?
Yeah. So the blood supply to the brain, even more so than the glia, I think generally got ignored for a very long period of time. And the reason why I think that this happened is because, again, of how we study the brain. So if you're taking sections of brain tissue to look at under a microscope, the way that you sort of like dehydrate and process and those brain sections, the blood vessels in the brain, which are small to begin with, like completely collapse down. Like all that's left is like a space where blood used to flow, and it's usually very, very small. So it's easy to ignore. And I think that's one of the reasons that it has been ignored. But if you think about in life, somewhere between 3 to 5% of the volume of the brain is blood. Each neuron is only like 10 to 20 microns, which is sort of like the 10th of a width of a hair from a blood vessel in any direction. And the brain has around 400 miles of capillaries. So the way that I think about it is that all those cells that we talked about are essentially seeded inside this incredibly tight network of blood vessels that kind of allow the whole thing to function in the first place. And this is important because, any time a certain part of the brain becomes activated, it needs oxygen, it needs some kind of energetic substrate, it might need nutrients to run that process. And that's going to have to come through the blood supply. So this is idea of neurovascular coupling, which is that as neurons and local astrocytes get activated, they release things that dilate the blood vessels nearby. That diverts blood to that area, so that there's, again, this like matching between supply and demand, or to help support capacity or function. And this is really critical as we get older. So there's a huge overlap between vascular disease and dementia risk, or what we see in the brains of individuals who had dementia. So if you think about the different categories of dementia, the most common is Alzheimer's disease, which is maybe sort of 60 to 80% of dementias. The next most common type of dementia is vascular dementia, so specifically due to like small clots or dysfunctions in blood vessels into the brain. And so between them, Alzheimer's disease and vascular dementia make up something like 70 to 90% of dementias. But there's actually huge overlap between the two. So the majority of individuals who have Alzheimer's disease have evidence of vascular disease in the brain. So there are many people who think that maybe it's an artificial separation between the two. But when we think about risk factors for dementia, some of the strongest are related to cardiovascular risk and or metabolic health or metabolic disease. So blood pressure being one that we know is a significant risk factor for dementia. If you treat high blood pressure, you significantly decrease dementia risk. But then other cardiovascular risk factors matter as well. So blood sugar being one, blood lipids, cholesterol definitely playing a role too. And more broadly, people often say like what's good for the heart is good for the brain, and cardiovascular disease risk factors and dementia risk factors certainly seem to track together. And that's probably because the process that's happening in the arteries of the heart, in the coronary arteries, the process of developing atherosclerotic plaque that can then rupture and cause heart attacks, that same process is probably happening in the blood vessels in the brain. And so if it's if the symptoms or the issues develop in the brain first, then that's going to translate into cognitive decline. And like I said earlier, there are some parts of the brain that are particularly susceptible to that, such as the white matter. So there's this really intimate connection between the blood vessels of the brain and how they function, and then also how that translates to risk of cognitive decline and dementia later in life.
So after taking a deep dive into what we know about what the brain is made of, you wrap up part 1 of the book by looking at what a brain is actually for. And you argue that our brains are capable of adapting and improving at almost any age, but for that to happen, we have to provide our brains with the right environment. So one place to look for evidence that dementia is not predetermined is the indigenous population of the Chimane, who live in the tropical forests of the Bolivian Amazon. And they have some of the lowest rates of dementia in the world. So what's their secret?
The Chimane and some of the other Amazonian Amerindians are thought to have some of the lowest rates of dementia in the world, where dementia is almost unheard of, despite the fact that they do regularly live into their 70s or 80s, which is an important kind of part of that. So in general, it's thought that this is then based on their environment and the lifestyles they lead, which I think can maybe, again, give us a framework for how we would apply that to ourselves. So this includes day-long, physically demanding labor for hunting, fishing, gathering, and farming. It's estimated that even late into life, they have sort of 4 to 7 hours a day of continuous physical activity. They then also, at least as part of that, and probably, and to kind of lead into that, they have a diet that's rich in vegetables, fish, lean meat. When they look at, risk factors for cardiovascular disease, they have very low levels of heart disease. Diabetes is very rare. High blood pressure is very rare. So I think that combination of, physical activity, diet, and then sort of like the resulting difference in terms of cardiovascular risk or metabolic disease risk compared to, say, American and European populations, that's kind of been one of the main thrusts of research to suggest why they have very low dementia rates.
Tommy, as I recall, they also have a slightly, not dramatically, but a somewhat lower prevalence of ApoE44 and even 3,4. Have you seen that?
I haven't seen differences in the prevalence, but I have seen there are data from that group suggesting that those who do have ApoE4, in some ways it's protective.
Yes.
And it's protective particularly because they have a high parasite burden. And so ApoE4 is important for regulating the inflammatory response. And so those who have ApoE4 in those populations don't have an increased risk of cognitive decline and dementia. It may actually even be protective in certain scenarios.
Right. I saw that too. The thing I was looking at seemed to show it was slightly underrepresented, but you didn't have the same apparent consequence of the ApoE4. And, it's been thought for years the ApoE4 is protective in certain environments where you have a high infection burden, inflammation burdens, other kinds of issues.
Yeah, so I think that the Tsimane are one of many groups around the world where ApoE4 does not increase the risk of dementia, which I think sort of increasingly lends credence to the idea that the increased risk of dementia in those who have ApoE4 in sort of modernized, westernized populations is at least, I would guess, largely driven by an interaction between genotype and the environment rather than some baseline increased risk caused just by the gene.
Do you remember not long ago, maybe 6 months ago, we discussed informally a paper in which it was promoting the notion that ApoE44 is a complete determinant of developing Alzheimer's and should be classified in a separate way. Do you remember that discussion?
I do, and that is this is a paper published in one of the Nature journals.
That's right.
And I was actually several people were quite scared because of this paper, and it definitely made me very frustrated because even when you looked at their own data in terms of the development and trajectory of dementia in individuals who are ApoE4 homozygotes, it looked very different from the conditions that they were trying to say it was comparable to, like the mutations in the presenilin genes or
exactly.
the amyloid precursor protein genes. So there were a lot of people who were very scared, people who had copies of ApoE4, when in reality, again, like the burden of evidence suggests that it's really driven by an interaction with the environment rather than some like fixed thing based just on genotype.
Yes, we discussed it because we received so many questions about it on STEM Talk. We went into it in some detail in one of the AMA episodes, one of the Ask Me Anything episodes, and you and I discussed it beforehand to get your views on it, but it seemed like a rather harmful paper the way it was presented.
Yeah. Agreed.
So, Tommy, speaking of dementia, since the aim of your book is to help people improve their cognitive function and also reduce their risk of dementia, including Alzheimer's disease, it would be great if you could give listeners just a brief history of Alzheimer's disease.
I think one of the best accounts of the history of Alzheimer's disease is actually in a book written by Carl Harrop, who Ken and I interviewed on STEM Talk recently. So if anybody who's like really wants to get into the history of Alzheimer's disease, I encourage them to read his book. But I do think there are some interesting parts of the history of Alzheimer's disease just that are interesting both in terms of how they shaped how we study Alzheimer's disease, but also just because they're interesting. So Alzheimer was a psychiatrist who looked after patients in an asylum, and then several of them who he felt had some kind of dementing process, so they lost significant cognitive function, were unable to look after themselves on a day-to-day basis. He then looked at their brains under a microscope, and he actually first started to do this in 1906, which was the same year that Cajal and Golgi won their Nobel Prize. So, as all the stories go, Alzheimer's index patient was a woman called Auguste Deter, and she had been institutionalized by her husband after he felt that her sort of behavior and memory were declining. And when Alzheimer looked at Deter's brain under a microscope after she died, so he was her psychiatrist for I think about 5 years prior to her death, he saw in her brain collections of proteins. First, one collections that he called miliary deposits, so they look like millet seeds. These then became known as amyloid plaques. And then sort of like the tau tangles, or the tangled tau tubules that make up part of the axons of neurons, they get sort of disrupted and start to tangle together. And so like these were became the classical hallmarks of Alzheimer's disease. He worked with a small number of individuals who sort of followed a similar process, right? So in their 30s to 50s, they had some kind of severe, unusual dementia. And, depending on which account of the history you read or believe, he was certainly interested in these, but it wasn't necessarily him who classified them together under this idea of Alzheimer's disease. That was supposedly done by his mentor, Emil Kraepelin, who wrote it in a widely used textbook. And at the time, Alzheimer's disease was cases of unusual, severe dementias that present early in life. So that's kind of how it started, sort of in the early 20th century. And then for a long time, that's all Alzheimer's disease was. And then what we think about as Alzheimer's disease now in sort of older individuals was called senile dementia. So the dementia that happens in individuals over 65. That's technically what senile refers to, or old age dementia, age-related dementia. And then at some point in the 1970s, they were kind of, these two different groupings of Alzheimer's disease were brought together. So at that point, we had early onset Alzheimer's disease, which is generally caused by individual mutations in genes, like I mentioned, like the presenilin genes or the amyloid precursor protein gene. They cause early onset dementia, a fairly predictable decline in individuals starting in their 30s to 50s, and then late onset dementia, which is much more heterogeneous, much more related to the environment and lifestyle, although it does have a genetic component. But they were kind of brought together because they look similar under a microscope. You see amyloid and tau in similar ways. Even for like a couple of decades after that, people were still focusing on other things in addition to amyloid and tau in the field of Alzheimer's disease. Thinking about inflammation or other things that might be important. But then, at least for a period of time, as the story goes, there was such a focus on amyloid and then preclinical Alzheimer's disease, so like amyloid that accumulates in the brain before people experience cognitive decline or any symptoms, as a therapeutic target for drug development, that really all the other ideas of what the other things that might go into cognitive decline in Alzheimer's disease were kind of sort of put on the back burner. There's now been a reinvigoration of that because largely of the failure of many sort of anti-amyloid therapies. And so people are now more interested in the other cells in the brain, they're more interested in inflammatory processes and other things. But for a period of time, we kind of were really hyper-focused on amyloid and the amyloid cascade hypothesis, where amyloid accumulates, damages, stresses neurons, causes tau to hyperphosphorylate and become tangled, and then that sort of drives the loss of function of neurons and then eventually neuronal cell death. One thing that I found particularly interesting, sort of looking at the history of Alzheimer's disease, is that August Derter, the index patient of Alzheimer, probably didn't have Alzheimer's disease as we now think of it. So people have gone back and looked at sections of her brain that were in his collection. And first, the group thought that she had a mutation in the presenilin 1 gene, which is a genetic cause of early onset Alzheimer's. But then another group went back and they couldn't find it. So we don't think she had early onset Alzheimer's from a genetic standpoint. People have also looked at her ApoE genotype, and she wasn't an ApoE4 carrier, so she didn't have sort of like a big genetic risk factor for late onset Alzheimer's. And sort of the accounts that I've looked at, one of the most sort of consistent ideas is that she might have had neurosyphilis, which causes amyloid accumulation in the brain that looks very similar to Alzheimer's disease. And this doesn't like change the fact that amyloid and all that stuff is certainly part of the process of developing dementia. It's not everything, but it is certainly part of it. But I think it's just interesting that Alzheimer's first patient who everybody talks about may not have had Alzheimer's disease at all.
That's fascinating. Constantly we hear to stand by that in the near future there will be just an overwhelming number of folks developing dementia. You hear that all the time. And much of that estimate or that prediction is based on the notion of longer life expectancies leading to more older people worldwide, therefore leading to more dementia worldwide. And in much of the world, we've become quite good at treating infectious diseases and also better at treating and preventing heart disease, which of course is a leading cause of death. So if you do that better, you extend lifespan. Now, you point to a couple of studies that show similar trends may also be happening with respect to dementia prevention, and that is really interesting. There was a recent longitudinal study in the UK that found age-specific incidence of dementia had decreased. A later meta-analysis of studies involving people in France, Sweden, Iceland, the Netherlands, the UK, and in parts of the United States found that the likelihood of being diagnosed with dementia at the age of 70 is now lower than it was 20 years ago. So could you tell us a little bit about these studies and how you interpret them? Because this is really encouraging news and seems to run against a strong narrative.
Yeah, this was pointed out to me by Professor David Smith. He used to be department chair of pharmacology at the University of Oxford. And he did a ton of the foundational work looking at nutrient status and dementia risk, particularly B vitamin and omega-3 fatty acid status. And sort of he sent me some of the initial papers, and then these meta-analyses came from them. But there are two competing ideas that are both correct, but it just kind of, it's important to understand what they really mean. So, it's projected that dementia burden will double or triple in the next few decades. Potentially becoming the first or second, the leading or second leading cause of death in most, sort of westernized, modernized societies. In the UK, it's already the leading cause of death. In the US, it's still heart disease. And this is largely because, as you said, people are getting older. And so if we're better at treating other diseases, then that makes it more likely that we'll develop something else like dementia. So that's true. But what we mean when we say that age-specific incidence is decreasing means that at any given age now, you're less likely to be diagnosed with dementia than you were 20 years ago, for instance. So like you said, at age 70, you're now less likely to be diagnosed with dementia in the US than you were 20 years ago. But you still have an overall increased lifetime risk of dementia because you're projected to live longer. You'll just get to, if you get dementia, you'll get it later. And the reason why there are a number of things that have happened that I think explain some of this trajectory. So in that big meta-analysis, the first thing, or the thing they focused on, was cardiovascular disease. So we already talked about the fact that cardiovascular disease and dementia have overlap in terms of risk factors. And we've become much better at treating and preventing heart disease. And at the same time, we may be having that same benefit for the brain. And so that was one of their primary hypotheses. And one of the reasons people think that is that in some of the studies, so like in the UK study, the benefit in terms of decreased age-specific incidence of dementia was greater in men than in women. And we know that cardiovascular risk is greater in men than in women. So they thought that maybe that was driving it. But that wasn't the case across all the studies. Other studies have shown that, so say the Framingham study in the US has showed that since cohorts of people born in the 1930s and 1940s, the size of our brains, so brain volume, has increased, and at the same time, dementia risk has decreased. And this is like the idea of brain reserve, which is part of the more brain you have, the lower your risk of dementia. And so I think some of that, again, sort of across multiple different strands of evidence, I think some of that is because, as we moved out of the Second World War and the Great Depression, we got access to better food and more calories, and then that allowed more resources to be invested in brain volume. You can take that too far, so now we're going the other way with high rates of metabolic disease and obesity, which then have the opposite effect. But I think energetic and nutrient availability was still a constraint on brain health and brain function in the sort of middle of the 20th century. So that's part of it, because those individuals, they were getting dementia in their 60s and 70s and the 90s and early 2000s, and those sort of made the core of the data of what we thought about dementia. Then there are other things that have happened in parallel that may be having a benefit. So we've seen increasing access to education, particularly for women in the US and around the world, that may be changing dementia rate trajectories, because we know education is a significant risk factor. Similarly, like in the US, there's a big expansion of women in the workforce. So we know that complex cognitive stimulating jobs can be protective. So I think all of those things are maybe contributing to it. But we then have to balance that with an ever declining metabolic health of the population and the sort of associated dementia risk because of that. So some people are suggesting that we've kind of had this benefit at the population level, but we may be now going in the other direction. But the sort of main takeaway for me is that if you make these big population-based changes across those different areas, you can have a significant effect on dementia burden and dementia incidence, which then gives us hope that this is something that we can continue to work on and improve as long as we sort of understand it and kind of apply it at the population level.
That's a great reply, Tommy. So as we mentioned in our introduction, today's interview covers part 1 of your book, which addresses what we thought we knew about the adult brain being fixed and inevitably declining over time, and how we now know that's not the case. And in doing so, we've just had this really interesting discussion about what the brain is made of and capable of. So in part 2 of our interview, we'll be talking about what each of us can do to stimulate neuroplasticity in our adult brains.
You write that the recipe for changing and improving the adult brain may not always be easy, but the recipe is actually quite simple. Can you touch just lightly on some of the key topics that we will be covering in part 2 of your interview?
Absolutely. So I think that kind of the core ideas that we've talked about here kind of tell us that we've either sort of overcomplicated the brain or we've oversimplified it in terms of thinking about what causes Alzheimer's disease and dementia, for instance. But the core idea is that if you zoom out, we can understand that there are these conserved biological inputs that determine how well the brain and the body functions. And we can use that to kind of apply this in our daily lives to improve brain health and decrease our risk of cognitive decline. So I break this down into different chunks. So like the strategy, I call it, is to build headroom, which I have taken from a previous STEM Talk guest, Art Devaney, who's an economist who talked about evolutionary fitness. And his idea of building headroom was just how do you build maximum capacity in the system? And I talk about it in terms of cognitive headroom, Because if you can build additional capacity, then if you do start to lose some capacity later in life, you still have plenty of additional function left over. And the way that, the tactics that I use, within the strategy of building headroom, all the different parts of, lifestyle that and the environment that we're probably familiar with. So I have chapters on how to approach movement, nutrition, cognitive stimulation, social connection, stress management, and that's like enhancing capacity as well as reframing what it means to think about stress, sleep and recovery, and then other like common health and environmental factors. And then I sort of bring them all together into this model of how I think they work in a way that can help people apply it in their sort of, day-to-day lives.
You mentioned the late, great Art Divani. He passed on early this year in 2026. And he was, I think, a several-time guest on STEM Talk and a very interesting gentleman. He was a professor. He also played professional baseball. And he was sort of a different kind of person than one meets in academia now. You wouldn't see him in a safe space screaming at the wall or something. He was sort of a robust individual that really regarded life as a wonderful buffet to be devoured and to leave nothing left. And so Art, I think, made it to 89 years old, and he had a wonderful life.
So, Tommy, we're really looking forward to part 2 with you. So thank you so much for joining us today, and we look forward to speaking with you in a couple of weeks.
Thanks so much for having me, and I really look forward to the next part as well.
Thank you.
STEM Talk.
STEM Talk.
STEM Talk.
STEM Talk. STEM Talk.
So Ken, I always learn so much when we talk to Tommy, and much of my research over the years has focused on the brain and the challenges our brains undergo in extreme environments. And what I've learned and what Tommy's research and book and today's interview reiterate is that our brains are capable of so much more than we first realize.
So true, Don. There's no question that our bodies and brains eventually will slow down with age. But as Tommy points out, our brains are capable of so much more if we only will broaden our scope and move away from traditional assumptions about neuroscience and about the adult brain that have, in a sense, held us back for a long time. I received an advance copy of Tommy's book, and it is unsurprisingly excellent. I would urge our listeners to go to Amazon and pre-order it. Part 2 of our interview with Tommy focuses on the second half of this new book. Again, the title is The Stimulated Mind. And in this interview, we will provide our listeners with a recipe for maintaining and improving their brain health. It, too, will be a fascinating discussion.
I'm very much looking forward to that part 2 interview, and I also will foot stomp and support that excellent note about Tommy's book. We got an advance copy, and it's just fantastic. So this is Don Carnegie signing off for now.
And this is Ken Ford saying goodbye until we meet again on STEM Talk.
Thank you for listening to STEM Talk. We want this podcast to be discovered by others, so please take a minute to go to iTunes to rate the podcast and perhaps even write a review. More information about this and other episodes can be found at our website, stemtalk.us. There, you can also find more information about the guests we interview.